Good afternoon, everyone. I'm Ziyu Wang. I'm an MPhil Advanced Computer Science student supervised by Professor Pietro Liò. And today I'm excited to share my master's thesis on hierarchical protein representation learning via topological deep learning. I'll guide you through the motivation behind our approach, the novel data structure and neural networks we will develop, and the breakthroughs we've achieved. So today I will mainly cover four aspects. I will begin with providing some biological background of proteins, followed by what is protein representation learning and why it is important. Next, I will discuss the gap of the current methods in learning hierarchical protein representations. The way to achieve hierarchical representation learning is using topological deep learning. And since this is a very new field, so I will provide some background for the key domains of topological deep learning, as well as topological neural networks. After I provide you with enough backgrounds, I will share the details of my MPhil thesis and explaining how I achieved hierarchical protein representation learning with topological deep learning. And I will mainly focus on the novel data structure, protein combinatorial complex, and the new models we've developed. So now let's begin with the biological backgrounds of proteins. Proteins are sophisticated molecule machines that perform every essential functions in living organisms. Proteins can provide structural support. For example, the protein collagen hold us together, forming scaffold or bones, skins and blood vessels. And proteins can also catalyze chemical reactions as enzymes that accelerate chemical reactions. Moreover, proteins can function as antibodies, which are Y-shaped defenders that recognize and neutralize the invaders like viruses. Proteins also can do many other more things like controlling cellular communication and regulating genes. So given their central role in every biological process, proteins represent critical target for biomedical research. From a chemical point of view, proteins are by far one of the most structurally complex and functionally sophisticated molecules known. Starting from the bottom up, we have atoms. They are linked together by covalent bonds to form amino acids. Amino acids can connect with each other to form peptide chains, and we call amino acids as residues if they are part of the chain. The peptide chain will fold into a conformation that achieves the lowest energy possible. Although the overall conformation of each protein is unique, two regular folding patterns are often found within them. They are referred as secondary structures, and they are like this helix shape shown here, and also this shape shown here. So although those, like for most of those protein found in living organisms, more than half of the residues will fold into such structures, and the remaining will become the loops or coils that connect those secondary structures. And moving up the hierarchy, we encounter motifs. These are small recurring structural patterns that combine secondary structure elements. For example, like this helix turn helix shape here. It consists of two alpha helices with a loop connecting them. This motif often have specific functional roles like DNA binding or metal coordination. Next, we have domains, which are independently folding units that represent discrete functional modules. At the territory structure level, we see how all secondary structures, motifs, and domains within a single polypeptide chain arrange themselves in a three-dimensional space. This is where the protein achieves its final functional form. And lastly, many proteins don't just walk alone. The quaternary structure describes how multiple polypeptide chains come together to form hierarchical complexes. This hierarchical organization creates profound implication for machine learning, and each level introduces distinct type of dependencies and constraints, from local chemical interactions to global assembly rules. This suggests that we need architectures that can capture patterns at multiple scales simultaneously, from atomic neighborhoods to domain interfaces and to interchain contacts. Now, let's discuss protein representation learning. I believe that most of you have already seen the impressive work of AlphaFold, which predicts millions of protein structure just based on their sequence and evolutionary information. Up to date, we already have 200 million of protein structures stored in the AlphaFold database, which is 1,000 times more than the experimentally determined structures stored in the protein data bank. And this is still not the largest data set we have. For example, like the ESM Atlas, for example, like they have more than 3 times more protein structures hosted compared to AlphaFold database. However, even if we can generate millions of protein structures here, they are meaningless if we don't know their properties. And clearly, it is almost impossible to annotate all of them just by experiments. So how to address this problem? The solution is protein representation learning. This is a deep learning method that makes sense of the annotated protein structures. So basically, a protein representation learning model is an encoder that accepts protein sequences or structures and then convert it to a vector representation. And this vector representation can be passed to a decoder model that can be used for downstream tasks such as predicting gene ontology labels, antibody developability, classifying protein folds, or many more. And the two most popular ways of protein representation learning are sequence-based language models and structure-based GNN models. The protein language model accepts amino acid sequences and uses transformer-based architecture to get the embeddings for each residue. The GNN models, on the other hand, accept a 3D protein structure, for example, a PDB file. Then it converts the 3D structure into a 3D geometric graph, where nodes are residues and edges are interactions or the proximities of each residues. And finally, the GNN layers are applied to the 3D graph to learn the embedding for each residue. So protein representation is already widely used in the industry, as you can see from these news headlines here. Large pharma companies are collaborating with tech companies and academia to develop incredible protein representation learning tools for drug discovery. And there are also AI companies like Chai Discovery that achieved unprecedented success rates when designing fully novel antibodies. Although the current protein representation learning model are achieving satisfying results, they still face key limitations. For example, the current protein language model learns from residue information, and the current GNN models learns from residue information as well as their proximity and spatial relationship between a pair of residues. However, they all miss a strong biological inductive bias from protein secondary structures. And so why it is important to learn from secondary structures? So I will give you a very clear example to explain here. Like you can see this stick graph that illustrates the structure of the green fluorescent protein. This protein serves as a genetically encodable fluorescent marker and is widely used in cell biology. Just based on the stick graph, we could only see that there is a green chromophore at the center of this protein. And it's quite difficult for us to infer the exact properties from this stick graph with like hundreds of atoms here. By contrast, if we use a ribbon graph of the secondary structure arrangement of this protein, it would be much easier to understand its properties. For instance, the barrel-like structure created by the beta sheets clearly shows a hydrophobic kernel around the chromosome, and it prevents the water from quenching its excited states and thus ensures the stability of the green fluorescent. Additionally, you can also see loops which are ideal regions for modifications. Those areas can accommodate insertions of foreign peptides or domains without disrupting the core fluorescent properties, enabling the creation of fusion proteins and biosensors. So see, it's much easier to analyze protein based on its secondary structure instead of just focusing on residues. So for biologists, it is also true that proteins can be more easily understood based on secondary structures and their arrangements rather than directly analyzing the residues. For example, the biologists use secondary structure to classify proteins, like the popular protein structure databases like Cas and Scope. They all employ a 4-level hierarchy to classify proteins. Like 3 out of the 4 levels of the Cas datasets are based on secondary structures, and half of the levels in the Scope datasets is based on the protein secondary structure and the arrangements. So we can easily see that secondary structure composition and arrangements are very important for biologists to analyze proteins. Additionally, the use of protein secondary structure arrangements has been also proven effective in generative models. One impressive evidence of that is the Prot composer work, which is accepted to this year's iClear as a spotlight paper. This work uses 3D ellipsoid layouts annotated with secondary structure to improve designability, novelty, and diversity of the protein generation. As we see that the inductive bias from protein secondary structure is so useful in protein generation, we would also wonder that how can such inductive bias be added to protein representation learning models? And one possible and promising solution is using topological deep learning. Since this field is very new and its potential remains largely underexplored right now, I assume that most of you here have not heard of the term topological deep learning, or at least have very limited knowledge about this field. So long story short, topological deep learning is generalized geometric deep learning, and topological domains we're discussing here are generalized graphs, and topological neural networks are generalized graph neural networks. The most popular domain of topological deep learning is hypergraph. This is a very easy to understand concept. Unlike the traditional pairwise graphs, the only difference that hypergraph have is that hyperedges allow you to connect multiple nodes instead of just connecting 2 nodes like the traditional pairwise graph do. And this allows you to model higher order relationships. However, hypergraph are still modeling flat relationships and cannot be used to model the hierarchical relationships in proteins. Another 2 main domains of topological deep learning are simplicial complex and cellular complex. They are able to represent hierarchical relationship beyond just node level and edge level. The figure here illustrates how hierarchical relationship can emerge here. Like we can start from a point cloud, which is a set of nodes, or we can call them a set of rank 0 cells. Then by connecting some pairs of nodes with edges, which we can also call them as rank 1 cells, we get a graph. Moving on, by attaching faces to edges that can form a triangle, and attaching volumes to faces that can form a tetrahedron, we now get a simplicial complex. Cellular complex is similar to simplicial complex, but it's more general. It allows faces to be attached to any rings formed by edges and volumes to be attached to any topological spaces that is enclosed by faces. Simplicial and cellular complex are proven useful in areas like neuroscience and chemistry, but remains more niche compared to hypergraphs. I think one of the reasons for this is that they have very strict requirements to the hierarchical relationships they can represent. Like, for example, they require that if a face wants to exist, all its boundary edges also must exist. And this is not always the case we want. Like in proteins, if we represent a secondary structure as a face, it will require all the residues in that secondary structure to be connected. And sometimes it would cause artificial edges to be introduced in our graph, and as a result, eventually leads to unwanted message flow. A better topological domain for protein representation is combinatorial complex. And this is the generalization of both hypergraph and cellular complex. Basically, you can see it as a hierarchical hypergraph, where each hyperedge have a rank, and the lower rank hyperedge can be contained by a higher rank hyperedge. And combinatorial complex does not require any cells to have boundaries like cellular complex do. So as a result, it offers a more flexible hierarchical set type relationship that can work better for protein representation learning tasks. And I'll discuss how a protein combinatorial complex would look like in later slides. And topological neural network, as said earlier, is a generalization of graph neural network. The most important and the only difference between them that you need to know here is that a graph neural network only allows information to be passed from nodes to edges and edges to nodes, but a topological neural network allows information flow from any rank to any other ranks. You may wonder that, are there any existing works that already use topological deep learning and in particular combinatorial complex? The answer is yes, and there are works that have been accepted to top conferences like ICLR, for example. Like in this year's ICLR, we can see EN equivariant topological neural networks, which is a topological generalization of the popular EN equivariant graph neural network. This work represents small molecules as combinatorial complexes, as shown in the figure in the top right corner. The rank 0 cells are atom, rank 1 cells are chemical bonds, and rank 2 cells are rings and functional groups. It demonstrates the state-of-the-art result in predicting properties of small molecules, but has not yet been tested on large molecules like proteins yet. So can topological deep learning be used for protein representation learning, and how effective would this application be? This is a research question that my thesis aimed to answer. So I have proposed the Topological framework, which includes protein combinatorial complex, topological neural networks for protein representation learning, and comprehensive evaluation across 4 popular protein representation learning tasks. Now let's delve into the method and results that I get from my thesis. Here's a very high-level overview of the Topological framework, and I will walk you through each step of its workflow. The first step is featurization. In this step, we begin with protein structure from databases such as PDB, CAST, and SCOPE. Next, we calculate a set of initial features according to the protein structure hierarchy from the residue level to the protein chain level. Here are examples of the features we can use. At residue level, we can have residue types, their central carbon positions, backbone torsions, and more. And at the proximity or interaction level, there are only residue distance and their direction features. These first two level of features are widely used in existing geometric GNNs for protein representation learning. And the next two higher level features are proposed in my thesis, and they are secondary structure level features, and they can be like secondary structure type, size, and the torsion between 2 or 3 consecutive secondary structure, as well as the angle between 2 consecutive secondary structures. And finally, at the chain level, we can have amino acid or secondary structure composition, chain size, and more. But all these features help deep learning models to learn the geometrical and topological arrangements of the elements in the protein structure. The second step of the protein framework, Topological framework, is the construction of protein combinatorial complex, which is the data structure we use to represent everything in the protein structure hierarchy. This step is very straightforward. We just set all the residue as 0 cells and their proximity as 1 cells. secondary structures as 2 cells and the chain itself as a 3 cell. For all cells at the 4 ranks, they all have their own features. And you may also wonder why we can't just use a graph with supernodes to represent such hierarchy. Well, I'll say that this solution is definitely workable, but by representing secondary structure as a supernode, like an individual supernode, instead of a set of nodes that we use for protein combinatorial complex, we lose a lot of important geometric information of secondary structures, such as like their shape and their spatial arrangements. And in the next and the final step, we employ a topological neural network that accepts a protein combinatorial complex and let their features to interact via message passing schemes and get the vector representations. The models can be trained via pre-training tasks such as sequence denoising and structure denoising, although it's not necessary. The more important part is training the model for downstream tasks such as inverse folding, antibody developability, classifying fold type, and more. With proper training, we can apply our topological deep learning model to unseen proteins and calculate their vector representations at each level of the protein hierarchy and infer their properties. Now let's discuss the models I've proposed in my thesis. I have in total proposed 3 models and I will start with the most powerful one, which is this topological complete perceptual network, or TCP-net. This figure here shows the architecture of TCP-net. The network processes both scalar and vector features with parallel pathways from rank 0 to 3. It starts with embedding the raw features such as raw features from each rank with separate TCP modules, which we will get into in the next slides. After the embedding layer, interaction layers are applied for message passing. The protein channel here, like shown in this dashed line here, is optional and can be replaced by applying a standard proline function to the final load representations. The little pink boxes here are normalization layers. So basically, we just start with message passing at edge level and the secondary structure level and the residue level and optionally at the chain level. So this architecture is very similar to the existing work GCP-net, but with extra support for nearly hierarchical representation at secondary structure and at chain level. And here is an illustration of an individual TCP module, which fuses geometric vector information into scalar features using 2 methods, which are scalarization and normalization. Here, like HS, here represents scalar features such as residue identity, and HV represents a vector feature such as residue orientation, and F here represents a localized reference frame. So I'm skipping the detailed explanation here to avoid going too technical, but at a high level view, this module lets the scalar feature and vector features to interact with each other in a non-degenerative way, and thus provide SE3 equivariance to TCP-net. And by SE3 equivariance, I mean that the model is equivariant with respect to rotational changes to the protein structure. So which also means that no matter how you rotate this protein structure, the outputs embedding are all the same. Now let's compare our TCP-net model with the state-of-art geometric GNNs. To benchmark TCP-net against existing work, we use the following 4 tasks. From left to right, the first one is inverse folding, which means that given a protein structure, predict all the residue identities. The second one is fold classification, which means given a protein structure, classifies gross secondary structure arrangements. And this task has 3 test splits from easy to hard. The harder splits have protein structure less in common with the train and validation set. The third one is gene ontology prediction, which predicts the gene ontology label of a protein. To maintain the computational feasibility here, we only care the cellular component annotations here. And the final one is predicting antibody developability, which refers to the likelihood that a therapeutic antibody candidate will be successful during the drug development and manufacturing. And this table shows our benchmark result, and we can draw two key findings from these results. From the fold classification test, we can see significant improvement across 3 splits, especially the hardest fold splits, indicating that TCP-net has improved generalization ability in recognizing the fold of a protein. And from the cellular component and antibody developability prediction, we can observe notable improvements with no sequence information, as shown in the left column of those two benchmarks, and comparable results with sequence information, which are shown in the right column for those two benchmarks. And this demonstrates robust performance of our model, even without the sequence data. So since our TCP-net is showing superior performance for structure-only protein property prediction, one promising use case of our model is in the de novo antibody design. A typical workflow of de novo antibody design involves 2 steps. The first generates a protein structure using a diffusion model, such as RF diffusion, and then pass this generated structures to a inverse folding model like protein MPNN to generate candidate protein sequences. However, this caused many of the structures that cannot be developed as antibody to be passed to the sequence design stage. And the sequence design stage can be quite expensive. For example, you may need to sample 100 to 1,000 possible sequences, and each of the sample sequences you need to check whether it can actually fold into the structure you want, and whether it's toxic, whether it's soluble, as well as many other more properties. So therefore, we would want a filter model that can predict properties solely based on the structure information, and use this between the diffusion model and the inverse folding model. And I think this is where we can apply our TCP-net model to. Besides TCP-net, I have also developed 2 other simpler geometric topological neural networks. I have developed GVP-TNN, which is a simple topological enhancement of the popular GVP-GNN. And I have adapted the existing ETNN work to protein representation learning tests as well. And I'm also skipping the detailed explanation here just for time, but just to let you know, they are very simple modifications of existing work that support topological modifications and message passings. Here are the results, and we can find that compared to TCP-net, GVP-TNN and ETNN are showing mixed results rather than consistent improvements. So the key takeaways here are, first, topological deep learning enhancement can improve protein representation learning performance, but requires careful tuning of the topological message passing architectures. And simple modification of existing work won't work. And second is that fold classification tests, which rely heavily on understanding secondary structure arrangements, see the greatest benefits from TNN's higher rank cell updates. So to conclude the contributions of my thesis, I have first developed protein combinatorial complex, which is a hierarchical set type structure to represent proteins that can keep all the geometric information of residued and secondary structures. And I also proposed TCP-net, which is a SE3 equivalent TNN for protein representation learning, and it achieves superior performance for structure-only predictions, which can be particularly useful for de novo drug design. And looking ahead, there are many ways to extend the current work. For example, we can integrate TCP-net with the protein language model. And we can do this by adding a vector quantizer after the last layer of TCP-net in order to achieve hierarchical tokenization of protein structures. And one big benefit of this is that TCP-net learns secondary structure in a non-degenerative way, keeping all the geometric information. So it is possible for TCP-net to translate residue-level tokens properly to secondary structure-level tokens, which can reduce the sequence length to the tenth of its original length. And this can be quite important for massively learning large proteins like antibodies. Additionally, we can experiment more about the impact of scaling up and pre-training our TCP-net. Right now, what I have done is training it with just 10 million of parameters directly on downstream tasks due to the computational limitations. But according to existing work, we can expect around 5 to 15% of improvement by scaling up and pre-training our model on a large data set like AlphaFold database. So here comes to the end of my presentation. Thank you all for attending and listening. Now I'm happy to answer any questions.